Mine's frozen too. Hello, everyone. Welcome to today's live broadcast, Microfluidic Single-Cell Exome-seq and RNA-seq Analysis of Tumor Composition. I am Brenda Kelly-Kim of LabRoots, and I'll be your moderator for today's event. Today's webcast is presented by labroots.com, the leading social media site for science professionals. For more information, please visit us at www.labroots.com. Before we get started, there are a few instructions. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers are always welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing your comments and questions into the box. We'll try to get to as many as we can, and we'll follow up if we don't have time today. If you want a better look, you can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, please let us know by clicking on the support button on the top right or use the Q&A button lower left. We'll make sure we try and resolve any issues. Now let's get right to today's presenter. We are proud to welcome Dr. Ionnasis Ragoussis. He is head of Genome Sciences at the McGill University and Genome Quebec Innovation Center, and associate professor in human genetics at the Department of Bioengineering. He is also an affiliated professor at the Biomedical Sciences Research Center, A. Fleming in Greece, and the King Abdulaziz University. He has developed expertise in all fields of genomics and functional genomics. His main interest is in developing NGS-based approaches to disease gene identification, as well as functional genomics approaches for the identification of prognostic markers and associated pathways in breast cancer. He is responsible for the genomic platform development within the McGill University Genome Innovation Center and Canada's Genomics Enterprise funded by CFI. He chairs the International Management Committee of the McGill University and Genome Innovation Center and oversees the genomic platform operation and new technology evaluation and integration. His lab works on technical developments, including the single-cell genomics lab and the mutation detection technologies with focus on the genomic and transcriptomic analysis of single cells. Supported by CF, the lab includes a Fluidigm Biomark HD instrument and 2 Fluidigm CI instruments, as well as an Oxford Nanopore MinION device. Both single-cell whole genome expression profiling, as well as whole exome protocols, are validated and available for external users. Please join me in welcoming Dr. Ragoussis for his presentation. Dr. Ragoussis?
Yeah, hello everyone. Good afternoon. I'm going to talk about the microfluidic single-cell exome sequencing and RNA sequencing analysis, concentrating in breast cancer tumors. I would like to thank you for the introduction, and I'll start straight away by saying that most of the data I'm going to show a result of a collaboration between my group at the McGill University and Genome Quebec Innovation Center and Morag Park's group, who is the director of the Goodman Cancer Center at McGill University. Further collaborators have been Sandana Batshu and Keith Sulbach at Fluidigm. Now, if we look at what we know about tumors today, is that if we need to apply sort of specific and targeted cancer therapies, we have to know what are the different subpopulations which consist a particular tumor. And what has been shown is that there is a lot of heterogeneity in terms of how different tumors in different persons look like. So different types of tumors in different persons are different from each other, but if we just go and see an individual tumor, we will see that there are different subclones or subpopulations of each tumor. And to make things more complicated, as we can see here, and to make things more complicated, if we zoom in to any one of these particular, what is seen as subpopulation that belongs to a particular clone, we can see that there is heterogeneity also within these populations, as can be seen by the behavior of different markers, either DNA or RNA markers. Therefore, one got the idea that we need to analyze these tumors at single cell level, and a lot of this has been developed by Navin just a few years ago. And the idea is that by applying single cell genomics, we can actually achieve the resolution that is required now to analyze the individual cells that make each of the subclones within the solid tumor, here as this example, and all the cells within the tumor. The next thing that is important to consider for single cell genomics is that we don't have only the tumor, but then we also have, as the tumor progresses, circulating tumor cells that can be analyzed in order to see how and what properties they have that allow a metastasis later on, and also disseminated tumor cells that can also give an indication about what drives the further metastasis and development of a cancer in the metastatic regions. In general, there are two ways to analyze the cells. So these can be isolated, as we said, either from the tumor or from a liquid biopsy in order to identify the tumor cells, or through bone marrow aspiration, for example. And these can be analyzed either now in terms of the DNA. In order to do that, we need to apply methodologies like whole genome amplification. I'm going to touch a little bit on that, or whole transcriptome amplification, in order to profile now the genomic and genome and transcriptome of the single cells, and guide further drug development or our better understanding of tumor biology in general. Now, in order to do that, there are two main approaches nowadays. One here in the top row, there are methods that will allow us to isolate any single cell from a population, and this can be done either by micromanipulation or serial dilution, flow sorting, or microfluidics, or laser capture microdissection. Now, on the other hand, individual and specifically rare subpopulation like circulating tumor cells can be captured through other types of technologies, like the DPRA, for example here, which applies dielectrophoretic cages, so there are low voltage currents that are used to manipulate the cells and drive them around and isolate them in a microfluidic device. There's also the magnetic mag sweeper, whereby little beads coated with antibodies against particular markers, cell surface markers, for example EPCAM, that are markers for CTCs, can be used to enrich for these rare populations from a big mixture and then analyzed. Obviously, these systems in row B can actually be combined with methodologies like in row A. The other thing that needed to be done was a development of technology that will allow us to analyze the transcriptome. And key here have been technical developments pioneered by, for example, by Richard Sandberg's group, by the Tang group, and Azim Surani and many others who developed highly sensitive RNA sequencing methodologies that allow us to actually generate a cDNA that reflects the transcriptome of single cells. And what has been key in these technologies was the idea that, you know, either if you here you use basically a primer that matches the poly A tail of the mRNA, and that fires up transcription. And the trick, the reverse transcription, and the trick here is to enrich for these transcripts and be able to isolate and sequence them later on. A key development in this has been the application of the smarter method. What is this? It has quite a peculiar characteristic, which is that we use again a primer that will start a reverse transcription from the poly A tail of an mRNA molecule, but by using MMLV reverse transcriptase that has a terminal transferase activity. Here, what happens is that once the enzyme finishes copying the RNA into the first strand of DNA, it starts adding a particular base at the 5' end, and the art here is to get an enzyme that will add the same base, like C in this case, at the 5' end. So several Cs are here synthesized, and the system allows actually now to incorporate a primer that ends in complementary Gs that will anneal with the Cs that have been generated through the terminal transferase activity, and immediately the enzyme jumps over and continues synthesis of the complementary strand from the 5' end. And that's where the name comes from. It's basically it means switching mechanism at the 5' end of the RNA template. Now, once you have the cDNA, you can now generate a library, amplify, and forward from sequencing. These methods and other similar methods that have been developed have the following characteristics. I'm trying to use an overview from a review of Peter van Loo and Thierry Wet. Here you can see that there are several methods developed that cover either the 5' end or the 3' end of the transcript. This can have some advantages if one is only interested at tags that will identify a particular transcript, for example. But smarter, as in this case, is a method that allows us to sequence the complete transcripts, and it's comparable to another method, PMA or SMA, and these methods use polymerase to amplify the RNA, or semi-random primer-based amplifications. They all yield comparable results. Now, the microfluidic instrument that I'm going to introduce has the following characteristic, just to put it in a map compared to other methods. It uses basically a system whereby you can visualize the cells. We'll see this in a moment, but it has the characteristic that it just requires something between 200 and 1000 cells. This is sufficient as an input in order to isolate up to 96 different cells. In comparison to other methods, it allows you to actually visually verify the cells, and it's possible to link it with downstream automations. But that's also possible with many other systems. Now, other technologies that have been developed include Droplet, published last year, for example, whereby now the cells are mixed with beads carrying primers with that are barcoded, and this method here on the top left has the advantage that one can now generate cDNA from a very high number of cells. So this method is suitable to characterize thousands of single cells. And commercial applications of this idea or similar applications include a system that is available from 10x Genomics, for example, and this is using the same idea of generating droplets that capture single cells and beads and perform the cDNA synthesis in the droplets, and that allows you again to analyze a very high number of individual cells. Other alternatives, I cannot cover everything, but other mainstream alternatives that are available commercially, for example, include the system from Wafergen here, whereby it's the same idea, but the cells are spread on top of a chip that has, again, primer combinations that allow you to perform locally cDNA synthesis and subsequent library preps, and it's also suitable for generating cDNA from about 1,000 individual cells. Now we have used the Fluidigm C1 system that captures 96 or more recently up to 800 individual cells for several reasons, which I'm going to go into, and try to make this work as a facility at McGill University. The good things about the system are that one can use this to actually capture between 96 and 800 single cells, and from there on you can perform either whole genome amplification and study the DNA or cDNA or RNA, which I'm going to touch upon a little bit. And from there on you can link this to either high throughput PCR instruments like the Biomark, which allows you to interrogate 96 samples with 96 assays, or you perform next generation sequencing using the cDNA or genomic DNA generated from single cells. Now the cells are captured within the chip, and what is the trick here? The trick is that the cells are captured in a very small volume, which is, as you can see here, 4.5 nanoliters initially, and then the cells are lysed as the next step, and a reverse transcription performed with this material in chambers that are increasing in volume, starting from 9 microliters and ending up in 27 nanoliters, ending up in 27 nanoliters, and then after the reverse transcription is complete, we have a pre-amplification reaction in order to amplify the cDNA further, and that happens also in progressively increasing volumes. But again, everything is kept in a nanoliter range, which achieves that there is a very good combination between high efficiency of the reaction and minimum contamination from outside sources, since everything is happening within a chip and completely enclosed. Recently this has been improved by reducing these volumes further to just a total of 26 to 27 nanoliters, and able to capture up to 800 cells in one chip. Now, after you generate the cDNA in the chip, the other important advance in order to generate efficiently material for next generation sequencing was the library preparation, and this library preparation is based on Nextera, which is a transposase based method. Basically, the cDNA is incubated with a transposase enzyme, which fragments the DNA and allows the immediate amplification of these fragments. And the beauty of the system is that these PCR products can immediately go and be sequenced in an Illumina sequencer. Therefore, this allows anybody to develop highly efficient and automated methods. It's a simple workflow, and it integrates also, the method integrates indexes that allow you to multiplex high numbers of samples and sequence them efficiently in terms of cost. Now, I will give some results from one example that we have investigated into some depth, and it shows here an approach whereby the Morax lab takes material from a tumor that is treatment resistant. It's a breast tumor. Then the tumor is propagated in a mouse model as patient-derived xenograft. And then the cells from the xenograft can be used as a culture, as an explant culture, or analyzed in more detail in tissue sections for histochemistry. And what has been shown before we went on to analyze the tumor further was that if you look at the primary tumor from which the sample has been derived and the xenograft, the histology is the same, and the characteristic of this particular tumor was that it was ER negative, PR negative, and had some HER2 positivity. So the system that was set up as a model system was to actually investigate the tumor and single cells derived from the patient-derived xenograft for RNA analysis and DNA analysis and protein analysis, and then obviously use bioinformatics to draw conclusions, but also to search for markers that will allow us to select subpopulations and then perform functional analysis of these subpopulations that would actually give us some information on possible ways to treat these resistant tumors. First thing that happened was to actually perform an expression analysis of the patient-derived xenograft cells, so these are the bulk cells, and compare them to the tumor. There are two samples from the tumor before and after treatment, and what you can see is that analysis of this data showed that this tumor is part of the basal breast tumor subtype, but it seemed to be a little bit different than the most of the other basal other samples that have been classified as basal. Anyway, what was reassuring is the fact that these samples all go together, and they seem to be therefore indicating that the PDX reflects as much as possible the original tumor in terms of expression profile. And that's also indicated here. This was further strengthened by using classification of the tumor based on PAM50 signature genes, and this shows clearly that the PDX and the primary is part of the basal like subtype. But basal, it's quite an issue. So basal-like is not just in order to appreciate the results. Basal-like is a heterogeneous group. Most of these tumors are basically triple negative, but it's not identical triple negative. So the vast majority is hormone receptor negative and HER2 negative, but there are within this group are also tumors that can have one of the hormone receptors or can have HER2 present as well, similar to the case that we are investigating. Now, the capture of these cells required quite a number of optimizations, and I'm showing you this here to appreciate the issue that actually, like many tumors, the cells are not all of exactly the same shape and size, but there is quite a distribution of the cells in terms of sizes, as you can see here. But that was the good thing about the fluid system, is that we could actually capture and visualize the cells that have been captured and measure them in terms of size, so that we can always go back after expression profiling and see what is the phenotype of the cells. The efficiency has been optimized to be quite good, and we are able to capture up to 93 cells from out of 96 position using this system. The next thing is that we perform the cDNA synthesis in the chip, and if we run the cDNA that has been generated, we can see, perform electrophoresis. We can see that it actually does contain high molecular weight cDNA. Then we perform the tagmentation. You can see here the shift after tagmentation, and we select then for our library the size that works best with the Illumina system. Now, in order to be sure that actually our single cell cDNA synthesis is as efficient as the bulk, we also sequence this the cDNA directly using a PacBio instrument, which is a single molecule sequencing instrument that actually can sequence up to 10 and 20 kb of DNA. And here you can see the bulk samples sequence, the average read length of our bulk samples compared to the single cells here. And what we can see is that indeed the single cells have a good distribution of long cDNA fragments, very similar to our bulk samples. Being reassured about that, we go on to perform the sequencing and then analyze the data, which required quite a complex approach that I'm going to go through a little bit here. But one needs to process the counts as usual, remove duplicates, and then use a suite of different softwares to actually analyze the data and normalize the data, which is quite challenging in this case. How does the performance of the reads look like? So here are raw data from single cell samples from this tumor, and what we can see is that if we sequence the bulk, so this is now something resembling 200 cells from this tumor, and we generate about 17 million reads for this, we can identify close to 20,000 different genes, which is close to what you would expect from the bulk samples. Single cells, we sequence between 5 and 7 or 10 million reads in this case, and what we see is that we identify in general something like 7,000 to 8,000 different genes from the single cells. The mapping rate is quite good, and you can see here that most of the reads are very useful, and it's above 0.9%. 0.9 is the mapping rate, which is very good. Now, how many reads we need depends on, it's a cost issue, but in general, this graph shows that with about 10 million reads, one should be able, and we worked out that we can capture most of the genes. For the single cells, it's a matter of cost, and in general one operates in this area close to probably 1 or 2 million reads, depending on what is the aim of the project. Now, how many reads you get is one thing. How well do they work in covering the different transcripts is the important question. And I'm showing you here how we cover transcripts from 5' to 3' end. And it's divided into low, medium, and high expressed transcripts. So basically what you can see is that in the high expressed transcript we get good coverage from 5' to 3' end. The method has been improved over the years, and we can see here very clear, nice results whereby the profile of the coverage is resembling what you would get from the high expressed transcripts, what you would get in a standard RNA sequencing using Illumina or other technologies. For the medium transcripts, you can see that there was somewhat a bias towards the 3' end, getting reads that map predominantly to the 3' end compared to the high expressed transcripts. But now, with quite a number of improvements, we can see that in general we don't get such a predominant bias anymore for the mid-range transcripts, but apart from a couple of exceptions shown here, for example, whereby you would then take note of this sample and see whether it should be removed from the analysis or not. For the low expressed samples, the coverage is still very good, so there is some 3' bias, but in general we still cover the 5' end, as you can see here, quite well compared to the 3' end. So the coverage is quite good, and this can be seen also if we project our data on the UCSC genome browser, for example. And here is a housekeeping gene that's not particularly important, but you can see that independent of how high or how low it is expressed, we cover all the exons of the gene. And here's a more tissue-specific gene. It's a keratin gene, and we can see again that we cover all the exons of the gene from the very 5' to the very 3' end. So the performance of the method is quite good in covering the entire transcripts, independent of their expression level. Next thing that you do after you analyze the data is that we take into account that we put in spikes. So we use 92 RNA spikes from ERCC, and these have different sequences and different relative ratios in terms of concentration to each other, as well as different lengths. And we use these spikes as controls, and what we want to see is that if there is a technical variation in the experiment from single cell to single cell. And what we project here is the levels observed for the bulk versus the single cells, and what we can see here is that the sum of the single cells correlates nicely with the bulk. And that's what you want. You want a correlation of 0.9 or so. The other thing that we do is to see whether there is, to clarify whether there is also another level of technical variation, and we look at the Pearson correlation of each now sample to each other, and plot the kernel density. And what you want is basically one peak in general that tells us that in terms of the ERCC spikes, there is only one population in what we have captured. And the next thing is to see whether the average, or if you add one single cell after the other, ending up here like 35 in this case, whether you will capture all the observations that have been made using the bulk. And in general, we want to be at about 100% in this case. And that's what we see. So this is the technical part, showing that in terms of cDNA synthesis, everything went well in the experiment. Now the next thing to do is to look at the mRNA expression, and what we can see here is we compare the bulk of the 200 cells to again the average of our single cells, and again we achieve a similar Pearson correlation as before, so it is around 0.9 to 0.8. Now on the other hand, we plot the Pearson correlation of each individual cell to each other, to every other, and look at the kernel density. And what we can see here is that in our case there is one main population, but there's a possibility that there is definitely one more population that is smaller possibly, or 2 or 3 different populations in terms of behaviors. Again, these behaviors could be technical or biological, but we can see here where this experiment can lead us into identifying a number of subpopulations. The other thing that we want to clarify is whether if we add one data from one single cell to the next, from 1 to 35, whether we will capture all the information that is in the bulk. And in this case, we see that we capture over 0.8 of 80% of what we can observe with the bulk. This is quite reassuring. So actually, it tells you that if it points towards being able to see populations that are about 10 to 20% of the out of the total number of cells. Then we use, in terms of analyzing the data further, we use the spikes and we look at the fitted values of the spikes, and it's most successful if you take their length into account, and then we use the spikes that show the best behavior, like here, to normalize the rest of the samples and the rest of the transcripts. Sorry. And if we plot here, we plot the variance versus the length, and the spikes can be seen in blue. So we perform this normalization based on the spike behavior, and we plot then again the coefficient of variation. This is the squared coefficient of variation on the y-axis, and the average normalized read count here on the x-axis. And this allows us to select basically a group of genes that show variation that has a good chance to reflect the biology. And in this case, we took about 500 to 600 genes, and what you can see is that indeed, if we use these nearly 600 genes, we can actually separate our single cells into 2, possibly 3 subpopulations. So once we have performed this analysis and we got this result, we do the following. We look at markers that are used to classify breast tumors into subclasses. And in this particular case, we use the Ames method, also developed at McGill and Hallett's group, that uses a classifier based on 150 genes. So we take these 150 genes and look at their relationship as published by the Hallett group, and see what subtype it's going to come up with. And what was the result is that our single cells could be classified into 3 groups. One was similar, sort of classified to HER2 as HER2-like, then basal-like, and some were classified as normal-like. And this is what we sort of suspected from the original tumor data, that there will be cells that could be classified by that, but it has to be done in order to be shown. The same we reproduced using single cells from the explant culture. We take the single cells from the explant culture, perform the expression profiling, and apply the Ames classifier. And again, we see 3 subpopulations: basal-like, normal-like, and HER2-like. So we immediately know that what we have observed during the quality control, that it is reflected in the behavior of the data, and that we do have at least and maybe 3 different subpopulations, or even more, but we can be sort of sure that there are 3 different subpopulations in our single cells. Now, in comparison, if we apply the classifier to our single cells and the bulk, that we had a couple of bulk experiments that we did, then what we see is that the bulk is always classified by applying the same conditions, is always classified as basal-like. And this is an important point as a take-home message. We would never have observed this if we did not analyze the sample at single cell level. So whatever subpopulations we take, they come up as basal-like once we use bulk cell analysis, even going down to 200 cells. Now, if we take the classifications that we have done, I showed you that there were 2 or 3 different subpopulations reflected here. What we have observed is that these subpopulations are enriched for different subtypes, and that can be seen here. So a bit better. So what we did was to clearly see what genes are correlating with the particular subtypes, and there are 2 groups of cells that we have here. And what we can see is that the one group here in red contains the normal and the HER2-like cells, and the other group contains mostly the basal-like cells. And their characteristic is based on high MYC expression or high EGFR expression. And we looked at EGFR because we wanted to have a cell surface marker that could be used as, according to the scheme I showed you at the beginning, to select the subpopulation. And in order to do that, we needed a cell surface marker that will allow us to actually get the cells and sort them using FACS technologies. So you can see that they separate nicely in these 2 groups. In order to confirm that, and this is this was done in collaboration with Fluidigm, we used an assay which is a multiplex protein extension assay that uses antibodies against 2 different antibodies against a protein target. These have 2 different oligos, and if they are in proximity, you can you get binding of the overlapping or matching sequence here, and then DNA, full DNA, is synthesized using an extension reaction. So once the DNA fragment is synthesized, when we apply qPCR to analyze the data. So we applied this using a panel developed for breast cancer, and we wanted to see whether the protein data will correlate with our expression data. And here you can see what we get. So actually, if we look at the MYC expression and the EGFR expression by qPCR, so we repeat the experiment, but this time we don't perform mRNA sequencing, but we perform high throughput qPCRs, including PCRs for MYC and EGFR. We can see that these 2 are negatively correlated, and the same here we perform with the at the protein level, and we get quite similar data in terms of qPCR. So then it was a matter of confirming, though, this result. So since what we have seen from our expression data was that there were cells that had high MYC and cells that had high EGFR. So I hope you can see here. I'm only showing you an example. So we look now at tissue sections, and if we stain for MYC and EGFR, what we see is that they stain distinct cells, and they are exclusive from each other. And that, interestingly, we went back and Morag's lab investigated also the primary tumors, and what we can see here is that independent of whether you look at the primary tumor or the patient-derived xenograft, you can see the populations of high MYC containing cells here and here versus the high EGFR containing cells, and they form distinct groups within the tumor. So we are happy to see that this was reproduced, not only what we see in single cells was not only reproduced in the PDX, in tissue sections from the PDX, but also from the tumor itself. And nicely, the data you see here in this experiment recapitulate what we see with a single-cell RNA sequencing in terms of expression of MYC, EGFR, and subtype classification. Now, what was quite interesting is that if we investigate what happens if this tumor metastasizes, and here is an example of an early metastasis in the lung, what we see is that this metastasis is negative for MYC and contains the EGFR cells only, so it starts, the metastasis at an early stage starts from the EGFR containing cells. This doesn't mean that the metastasis is driven by EGFR, but it's just it is a marker for this subpopulation of cells from this tumor. Now what happens next is that although at the beginning we have only EGFR cells, once the tumor grows, then it starts to be containing subpopulation that is now expressing high levels of MYC, which continues to be sort of exclusive of the subpopulation that contains the high EGFR. And what is interesting is that basically the metastasis here has now has started to recapitulate the subpopulations that make the original tumor. The same is observed when you grow the cells in spheres, so they contain cells that are either high in EGFR or either high in MYC. Then the idea was, okay, we can see this, but is it actually something that can be observed in other tumors? And indeed, here is an example out of 11 different tumors that have been analyzed at this level of detail. And you can see here an example of 3 tumors shown here, whereby we do have the same profile. It contains high EGFR or high MYC cells that are in distinct areas of the tumor and show a very similar distribution as our original index case. This is not always the case. So in the next slide shows all the other tumors that have been investigated. But what is interesting is that indeed the tumors can be split into ones that have MYC, some have EGFR and no MYC, and there are obviously tumors that have both. But my point here is that if you look at these tumors, performing expression profiling of the tumor will not allow you to distinguish between these two subpopulations. Now, since what we wanted to achieve was to have a cell surface marker to fish out the one subpopulation from the other, this is what was done, and the next step in experiments performed by Paul Savage in Morag's lab was to actually use FACS to sort the EGFR positive subpopulation, And to our surprise, this is the population that can grow in tumor spheres, while the negative population, which is the biggest population, does not grow in tumor spheres. And this experiment has been repeated several times, giving consistent results. And again, if we look at now in the animal, again, it is the high EGFR population that generates tumors that are quite big, while the negative population is quite inefficient, and MYC containing is inefficient in actually generating tumors. And that can be seen in this type of analysis here and here. So after this, we can say that indeed the method has allowed us to identify subpopulations that would not have been identified using bulk analysis, and that actually these subpopulations are functionally distinct and have different growth and metastasis properties from each other, and are now available for further functional characterization in order to see which compounds would be effective against which subpopulation. Now, in parallel, in the last minutes, I will cover the fact that you can use the same system to actually look at the genome of these samples, and this is being done through whole genome amplification. And after whole genome amplification in the chip, we perform an extera capture, which is again fragmentation based, and followed by exome sequencing. I'll just show quickly the data. So the method works quite well. Here are the control samples, the tumor and the xenograft, which we sequenced at a very high depth of at minimum 500-fold coverage, and the single cells shown here, which have been sequenced at a coverage of 39-fold. But if you put all this together, you end up with a 1,380-fold coverage of the tumor. The mapping rate, as you can see here, for the single cells, the performance of the whole experiment was indistinguishable between single cells shown here, or most of them, compared to a standard exome capture shown here. And if we wanted to look at, I'll just go to the next slide quickly. So we analyzed the data, and what we see is that we could identify quite a good number of possible damaging SNPs in the tumors, and 27,000 of those have been identified, of which most were not in dbSNP. And I'm showing this here in order to demonstrate that because we sequenced also the germline, all these were not were homozygous reference, the germline, and shown in the tumor. Obviously, we compare the different tumor samples before and after treatment with the PDX and then the single cell experiments, and we see nicely that these are overlapping entirely, and the PDX can captures the information that we capture from the PDX and the single cells reflects the information present in the tumor. And what is interesting is if you take the SNPs and you look at whether the single cell, the information that is found from the calls from the single cells capture the calls from the bulk, and you can see that this actually is quite effective, and we capture 100% of the bulk, whether this is 200 cells or unamplified genomic DNA from the tumor shown here, again with the single cells by sequencing about 81 single cells, we capture the information that is contained within the tumor sample. And we use this, in order to conclude, we use this information to cluster the single cells, and that's shown here. And indeed, we can see that we can identify 2 or 3 subpopulations, so the pattern we get is very similar to the pattern we get if we use about 600 genes according to their expression behavior. Then we take this data and project them to the RNA sequencing data. So we use our RNA sequencing data to discover expressed now mutations. And I'm showing you this to actually simply say the following, that these indeed may be split into different categories, into different subpopulations in terms of behavior, but this does not explain the clustering we have seen according to subtype based on the expression level. So therefore, so far, we don't have an indication that it was the actual mutations that drove the behavior of these subpopulations. It can explain why in the metastasis we have EGFR, let's say, the EGFR containing subpopulation, and then from that you can derive the MYC containing subpopulation. So in order to conclude, we have performed the expression analysis that allowed us to successfully identify at least 2 distinct subpopulations with distinct functional properties in terms of metastasis and proliferation. We also found a lot of genes, but this is less interesting, so I'm not covering that. I'm finishing by saying that we had no evidence so far that the mutations were driving the clustering we have seen with expression profiling, and this can be used now to develop treatments for these subpopulations. And I finish here by thanking all the funders. Thank you.
Thank you, Dr. Ragoussis, for taking time to be with us today and bring us this important information. Before we get started on our questions, which we already do have a few, I want to remind our audience how to submit questions. Questions can be sent via the green Q&A button at the lower left of the presentation window, and we will get to as many of them as we can. Our first question, for the PDX model, does the number of initial tumor cells transferred to influence the expression profiles? For instance, if 10 to the 3rd, 10 to the 4th, or 10 to the 5th during the propagation of the tumor, does that change the expression profile?
This can change. I mean, not all, and there have been extensive publications in Nature and Cell about this issue. It's not a general phenomenon that if you do the PDX out of 10 or 100,000 cells, that it will reflect the expression profile of the original tumor. So one issue is that it is a bit unpredictable up to what extent the expression profile of the PDX will reflect the tumor. In our case, what we can say is that there is a very good overlap between the PDX and the tumor, but it's not clear how many cells would be required in each particular case to achieve that. But it has been where you do a number of experiments until the PDX does actually reflect the tumor.
I see. Thank you. We have another question. Can you describe the challenges of using single-cell RNA-seq from explant culture?
One of the biggest challenges from the culture is that the cells have to be dissociated from the culture surface, and that brings with it quite a lot of debris. And one has to optimize it using different methods in order to dissociate the cells from the culture surface without actually carrying different bits and pieces from the surface itself. So I found this the biggest challenge in our case, the debris that comes with it. The other challenge is to get the explant culture right, because what we have observed is that the expression pattern of the cells in culture can shift from the original patterns observed in the xenograft itself. So one has to check everything twice or 3 times.
It sounds quite complex. We have another question. Will single-cell sequencing apply to epigenetic study?
Yes, it is possible. So there's intensive work going on to develop methylation analysis at single-cell level, and it is possible. But there is a lot of development going on, and there will be standardized methods to perform extensive methylation studies from single cells.
Great. We have time for one more question. An attendee says, thank you for a great presentation, and asks, can we use single-cell RNA-seq analysis for other not mammalian animals, such as chicken?
We have used, there is no reason why not. I think one has to use a cell capture system that is suitable for the particular cells, but I think, for example, the one we use has very wide applications. I think it gets tricky. It's not, if you go towards very small cells like in yeast, it's going to be difficult, and you use special microfluidic devices. But in general, the analysis described and the other methods like Droplet, etc., are very well suitable for analyzing any cell type.
Thank you, Dr. Ragoussis. That is all the time we have. I would like to thank LabRoots for making this webcast possible. Today's webcast is going to be available for on-demand viewing through November of 2016, and attendees today will receive an email alerting them as to when that webcast is available for replay. We would invite you to forward that email to any of your colleagues who could not log on directly today. Thank you again to Dr. Ionnasis Ragoussis for bringing us this information and making this webcast possible. We hope we see you again. Thank you very much.
It was a pleasure. Thank you. Bye.